Hi, everyone. Welcome to this second training, Fundamental Lab Skills. It is really important. There's like a couple of things that is really important for setting up a successful lab, like regardless what kind of experiment you're doing. So here is one. So this is what we're going to introduce in this training. Just some conceptual stuff. Well, not really conceptual, like conceptual plus practical that can help you be successful, like in all the experiment you're going to do. So there's some like notebook keeping, practical making, label, storage, and stereo technique. Okay, so notebook is about, we're going to talk about notebook code and some of experiment analyze, like contain your experiments, your analysis, your personal note about your questions and ideas. And the protocol will got like protocol, like going to talk about how to make protocol, and that's for safety and efficient. Of course, even storage about your safety, and also about how we manage your assets and resource. And lastly, like store technique, I believe at this point, everyone, all of you have been gone through the lab walkthrough, and I'm telling you like these a lot about in our lab, like a lot all our stereo technique and what we wear PPE is mostly because we don't want to contaminate samples. We want to protect samples more than ourselves because it is like less hazard to us than to the samples, because once the samples mess up, your experience also mess up. Okay, so here is notebook. I must it is really a fundamental skill for you. Like first, like everybody will be given a notebook, and then you can write all your notes on it. And it is a lab property, and it is the evidence your work, and it's a legal documents, and like just a story that long times ago, uh, Dr. Simons, who was like the cancer center's named after him, discovered a drug, like a composition that can like okay, so again, I don't know all the details, or I don't remember I don't, but anyway, he discovered something, and then he was able to at the end the pharmaceutical company take it, and then long times after, and then the BYU won the suit that like they should like all the profit from their drug should be given back to BYU, and Dr. Simon got the funding, got all the money back, and he was able to go through the lab notebook and see who was contributing to that project and like distribute the profit for them as well. So it is really important to record your work, and so and also like in old time, that lab notebook is really important. Like if like when two labs are competing about the same things, and they want to climb who was the first one to like discover the idea or the stuff, like the new discovery. And what they will do is they were going to compare the lab notebook. Okay, so it's just like a two-side story telling you that how much notebook is important. Like beside it help you record what you do and help you reproduce, like how do you reproduce your data, and like you can find your result easier. So like you're welcome to go to my desk and take a look at the examples of all my notebook. Like here you can see like the notebook that is on the side. It has the initial number of the number, like one the number of the notebook. Like I have four notebooks, like it is like 1, 2, 3, and 4. And underneath is like what I do for those projects. But that is for me. Like I will have four, and like most likely undergrad usually only have one, and depends on like what project are you working on. Like in our group, I mostly I like to use that notebook as per project. So if like, because a lot of time you guys might be working on the same experiment in the same time, and that will be more useful when it is documented in one project, like in one notebook. So it will be more like, okay, if you're doing a folding experiment, it will be documented as LL something, but you will take note, your personal note, in your personal notebook. Okay, so but we'll go over more detail when you start using your notebook, but here it's a conceptual of what it will be like. Okay, so what is going to be in the notebook? There will be of course, it will be a table of content. It will have things like the experiment you're doing. That is what you're going to make protocol for. It also include your data analysis for your result, and you can put your literature review, like some of the keynote on there. And most important, I want you to use your notebook as the note for our discussion for data. So a lot of time you're going to have a data set, and after we discuss, I will give you some key points of like our thought process, and I want you to document that, because that will guide you of like what questions you're going to guide you in your data analysis, and also help you to remember just like, okay, what I'm going to do for the next couple of days or the next period. What is our goal? And that can be your checklist. But anyway, just make note on what we discuss. That is also important. So I'm going to start to showing a lot of like examples from the notebooks. Okay. Here you go. So this is like a protocol site. So before we just show you that is how the cover of a notebook, and let's show you some content. So this a protocol. It has like the protocol on this side and the method, and then the other side has like your result and the detail, like the table and the graph you have. And then I give you to me like just so like we'll go into like how to make a protocol in later slide, but for the result, it will be your like raw data and your data analysis when you're plotting after it. And I really like to do something is like make a PowerPoint as you go, because you will find it is like really easy to just, okay, I have a figure, I have Excel table, I just put it into the PowerPoint, and then below I have like on the note side, like you can have, okay, where's the data source from? Like list the data files name. And then like some of your thought on like those figures. And then after that, you can print your PowerPoint note and then stick inside to the notebook. And just like you notice like most of the notebook content in our group will be typed for a couple of reasons, because it is editable and it is like easy to pass on. Like if we want to transfer it, like get date, like what's that called? We want to share the data in between like groups, or we want to share the protocol. Like I want to send a new protocol to you guys, and I can just like I have an electronic version, and as you go, you have that, and then we can add a protocol, like have some basement, we can repeatedly print it and as our guide in the lab, and we can take note on that. Well, later I'm going to explain more in the protocol sessions, but like, and we have access to those electronic copy and everywhere. And you might have questions like why we just don't go into like all electronic? Like still there's some like, I feel like there's still some value of print version, because I don't know, maybe I'm old school. Sometimes sometimes writing note is a lot faster, and that like writing will be valuable when you're doing your personal note, that like you don't necessarily care if other can read it after you're long gone from the lab. But like just as I promised, like sometimes I don't recognize my handwriting 3 months after I write it. So even just for myself, that of electronic like copy is much better than handwriting copy. But like why we print it out still, like sometimes I feel like if I want a quick answer, I can go through my notebook really easy and I can show somebody else a lot faster. And here's some of like examples of like the benefit of paper copy. Like we have been using this table and this table a lot, like in between like my group, and also like the group sometimes will reference to my notebook like for those recipe because it's printed out already. So you have easy access, and these are the like samples protein concentration, and that you will find we use that a lot later because that is like what we do in folding group just to get those concentration like from time to time. So it has an easy access for paper copy. Okay, so next thing is about like take note as you go. So notebook, just really straightforward. That is its name. So like that is where we can take note. So like as like one things about being a researcher and scientist is like we have to just be really observant about our environment. Like we need to observe all the changing in what we do. So as you do an experiment, I like you to observe the like all the details like about your experiment, like color change. Like if you're doing a folding experiment, you have iron, like it has a like iodine, and iodine's brown, and it should be in your sample somehow. And as the iodine goes, the color of your like liquid should change. And like to extent, and then another example is transferring is the iron bonding protein. When it is bound, it is like the solution looks red. And like this experiment that we did also ultracentrifuge, we spin it at super fast speed. Like so regularly our centrifuge we're doing like 14,000, 14 kg, and also line for 15 minutes or 30 minutes. That's like our routine in our lab. And when doing ultracentrifuge, like we're doing like 200 kg and overnight. So like that's really like strong spending like centrifuge force. So what we at the end, like you will see, oh well, the solution, like the iron all got poured down from the protein and the solution become clear and you have a red pellet there. And also like one typical color change is like in your BCA assay, protein concentration assay, that you are going to see your experiment color change from clear to green and then to purple. And so those are important indicators of your experiment. And you also want to like notice like if there's any precipitation like here in your experiment or in your buffer, if your reagent has precipitation, that usually indicate it has some issues. So you would like to pay attention to all those details and then record them. And nowadays it's really really convenient that we have phones. So just take a picture and then pick up use video, just include those details in your lab, like in your notebook, and your final conclusion of your notebook. And you're always welcome to use the printer in the lab. And then you want to print color, like those printers are also connected to the printer in the central room that will give you color printing. Okay. Okay. So and then so that another benefit that you can find is about project, like making project code, is you have printed version already, and a lot of time we're going to repeat our experiment over and over and over again, and with small modifications. And here we have two images that show you about take note as you go. So like you can see as like for this like this big one, we have a protocol printed that we already made it. But as you do your experiment, you take note of those details that is not quite the same as your plant, and like you can see there's a lot of messy change, and that was a disaster, by the way. I was doing that experiment, something went wrong, like as my expected, and then so I have a lot of notes taken based on that, about like I have modification as I go, and then And then, so I also have like the date and time of the recorder for the experiment. And that first it tell like it record when you do the experiment, and like it help others know what you're doing. That is especially important when all of us like in the group are doing the same set of experiment. Okay. And the other things about like taking note as you go, yes, about a timestamp. Okay, so it also help you estimate how long you're going to working for next time. Okay, and here is another note of like note about, okay, like you're doing a routine experiment and what are the changes you can do. So just like take as detail, the note should be as detailed as you can, like, and like really the more detail you have, it's help you reproduce your experiment better, and it helps you troubleshoot, and like it also helps you have a material for you for your presentation, like this picture I just grabbed it from my note before. Okay, so we're going to talk about notebook code a lot, and here just examples of notebook code. Like this is page 15, and as here you can see that is like LL, that is my notebook number 4, and starting at page 15. Okay, let's go to the next slide. So, well, notebook code, besides what we talk about experimenting your note protocol, it should have a table of content at the very front, so you'll save some space for your table of content. And I usually like to use like about the first 5 pages for a table of content. So just like in case I have big handwriting. Right, and yes, that is the part we have handwriting, I think. But you can do it as like a print out after you have 1 page, or you have label of content recorded somewhere. That's also good too. So talking about notebook code, that is really important because that links everything to your like what you do in your in lab. Okay, let's go through the like table of content first. Like there's a date, and you have your experiment name or your objective. Something like here, like I have reanalyzed that this pure data analysis, and like some of them, like these are all, this is a troubleshooting, and this is also data analysis. And then like we have experiment, and then this is the notebook code, and it tells you like, okay, the page of it. Okay, so what is notebook code? By the time like you should be really like want to know what is notebook code, right? I have mentioned over and over again. So notebook code has two it's a 6-digit code. The first 2 digits is your first and last initial. The second, like the third digit is the number, like notebook number. Like I have 4 notebooks, so you can see like I have like LL1 something and 2 LL4 something. Okay, and the last 3 digit is your page number, because the notebook we have in the lab is 3 page, about 120 something pages big. So you want to make it to be 3 digit. So an example, it will be like LL like 1006. Okay, so notebook code is the key. Why it is so important? Because it is key for your all experiment data. You use the notebook code to label your sample, your tube, your reagent, your raw files, mass spec file, like starting with notebook code, and your data analysis file. Your Excel file should also start with notebook code, because that help you like link all the things together, and in one notebook you should so if somebody are interested, okay, I want to see my result for TMT, and I know it is at like, okay, LL 1076, and I can use it, like use this code to find my data analysis file, find my presentation, and find the raw file if I want to reanalyze it. So, and also a lot of time, like in your protocol making, you mention, okay, I have my sample source is from like LL 1116. That is like a human serum intake. So like you know, oh, I can go to the LL 1116 to find the protein concentration and some of experimental detail for those set of sample source. Okay, and then so that is line over code is important. And so speaking about like it line over code with the experiment detail, I also need to talk about like file storage. So there's a lot of data analysis in our lab. So because mass spec data is huge, and then you have like we can analyze it after it is in a Excel format, and you're also going to run it in a secondary like couple of software to analyze it. So you generate a lot of raw data. So all the mass spec files should be stored in large array 2, and then and then also like when you're doing analysis, what you should do is you should like let's just say you should have your Excel file name after your noble code. Start with your noble code, and all the material like about the experiment has the same notebook code should be stored on like within in the same folder. And the folders, like if you're using your computer, your laptop, you should have like a folder about your like price lab research, and then like in under that price lab research, you will have like your like each of your noble code experiments. This is there, and then if you're working on a computer in the lab, like you should also have like such a folder of your folder, and under your folder, and there will be your experiment analysis and PowerPoint and particle and such like everything related to that notebook code. And just remember, like don't store anything as C drive, because like one if let's C drive like the computer have to be like rebuilt, then you lost any data that is in C drive. But if it is in the like stored at the, like not the system drive, if it is not stored in the system drive, you have some chance to recover it. Okay, also like you like want to upload your data periodically to this Synology drive. Okay, I am going to show you guys the large array. So just give me a moment that I'm pulling it up as my internet goes. Okay, in the computer room, all three computer have the address of large array store. That is a storage area. I show you guys where the SOP stored when you go through the lab walkthrough. So here, just like a large array 2, and it should have the password like already there. But if like when you open the large array that you cannot log in, just as undergrad to help you, like a grad student to help you. And then it takes a while to log in. But here as you log in, like you can see like there's like bookmark to it, and like Synology large array 2, LA2, large array 2, and these are just different way to access to the drive. Okay, I showed you how to log in here, but I think people have something pulled on here already. So this is something already pulled. So your mass spec file, like everybody will get a folder on the price lab. You should have a folder that is your name if you ever run samples there. And then like run mass spec samples under your notebook code. You should have a notebook under there. But like usually like, and if it's like just an example of all the voting data is under Lavender, and then it should be going to a, like that is for your massbed file. Okay, my massbed raw file is here under massbed raw file. Okay, so that's like when you get your like raw data. And here also has a undergrad analysis data. That is where you will store your analysis data, back it up. So you can see like there are different names, and you're just going to like periodically upload all your data up to the Synology too. Okay, so let's go back to the PowerPoint. Okay, so like list questions you have about it and just ask me. But I hope that is detailed enough for you to know that everything you do in the lab should attach to a notebook code. So it helps you to organize your data and also help people after you to know what you learned. Really, just I want to emphasize again, like we are here, even as a grad student, like a grad student is here typically for 5 years. And undergrad, like depends on what you do. Like you're here for like at least 1 year, sometimes like 3 years. And no matter how long you feel like it is, your project, like what you do, you want to keep it as a legacy. Well, not really like not legacy, but you don't want what you do, like you don't want to live and nobody will understand what you try to do after you're gone. So you want to like keep it as detailed as you can, as organized as you can, so people after you can continue work on what you have been like working so hard. That then you will not waste like all the effort you put in, even you did not finish, can continue in a meaningful way. Okay. So here we go about protocol making. That is a big deal. So I also emphasize a lot. So finally, we got to see how we are going to do that. Okay, so about protocol making. Why it is so important to make a protocol? I like to because it is about planning, about being prepared. And like being prepared and planning is so important in research work. I don't know if you ever like if you like to cook, and then whenever you're looking for a new recipe, you try to follow the recipe, and then after you have your recipe, you can you might be feel free to make some change because of based on the recipe, but you have to have something to base on. So that is like our protocol. And really, sometimes for chemistry, you can imagine it is just like cooking, like you're mixing different ingredients together and like incubate it for a certain time, and you have to control your temperature, and you have to look at how they change. So anyway, so planning is important, and it is important in many things. Like I like to see it as like you're going through a mess. There's so many things you can well, if you're going through maps and if you have a map of the maze already, then you can plan it out. Like you can look at, okay, that is the end and how I'm going to go through it. And then you just based on your planning, and you can go through the maps really super easy. And that is like, it is not saying that like research is not it's not saying research will never change and you can plan for anything. Like as I say, like a lot of time plan doesn't things doesn't go as what we planned, but at least we still need to be prepared, and then as long as we're prepared, we can deal with those change a lot better. So practical is about safety, it's your guideline, and it also boosts up your efficiency. I like for all the group that in people in my group, I ask you only going to wet lab to start your experiment once you already have your protocol, because I don't want you to waste your time in the lab to do the calculation and so those things that you can definitely do ahead of time. And so when you're in the lab, what you're going to what you do is just just like following your recipe and then following your recipe and then follow your recipe. Sorry. Okay, sorry about that. Baby yelling. Okay, so I just like a lot of time you waste your time, like the most time if you're not prepared, the most time you spend in the lab is searching for reagent and doing calculation and just getting ready for your experiment. So, like to do that more efficiently, I want you to do all those calculations and then like look like like planning for what you're going to do ahead of time. So that's why you have the protocol. So once you have the protocol, you take the protocol into the wet lab. You have all the recipe you need, and you know exactly what you need to. What you do is like just like take those reagents in front of you and mix them, and start, and then you can just like go and go and go, and that saves you a lot of time there. So and then it also help you plan your time better. Like a lot of time there's incubation time in between samples, and it is especially important to plan those time if we want to crash like power through a lot of sample prep and to get the data onto the mass spec. So particle is important, and again, and it helps you focus on the detail, not just like what are you going to do, but you can pay attention to like okay what's happening, and then like you can like as I showed before that you can take note as you go. So this is like why we want to have the particle, and in the next video I'm just trying to break it in half. So we're going to just talk about how like what is in the particle in a bit. Okay.